Hello, everyone. Good morning. I guess good afternoon for people on the east side. So I'm Rebecca. I'm a postdoctoral at the Center for Disease Control at British Columbia and at the UBC, and I'm a postdoctoral fellow here with this program. And I'd like to thank for the opportunity to speak about my projects here. And also, great talk, Min and Katrina. It was amazing work you guys are doing. It's really amazing how diverse is this program and how different types of projects we have. So my project concerns the modeling of surveillance systems, like to improve surveillance systems on the disease. So in here I have 3 projects that are following different. By chance, I'm following disease across scales. So I'm doing a project around wildlife risk, genomic risk, and public health system forecasting. I would like to start with the land acknowledgement. I live and work in BC, in Vancouver. That's the unceded territories of Musqueam, Squamish, and Tsleil-Waututh nations. And about me, I'm an applied mathematician, so my background is in mathematics, and I work with a lot of mathematical modeling, and currently I'm focused a lot on infectious disease modeling. And in this project, the fellowship is embedded within BCCDC, and within BCCDC, I'm collaborating with different service lines. So the idea, how it works usually is that we have the epidemiology intelligence services and the data science innovation team, and we can serve as like a consultant. Let's say we can do models, develop models and projects based on the needs of the other teams around. And then currently, I'm collaborating with the environmental health services and public health response within the respiratory team within the public health response and the public health laboratory. So just to give an overview of my work, as a mathematician, I'm going to be developing models to support and provide evidence for public health surveillance systems. So the idea is we can trying to improve the current surveillance systems or answer new questions that might come up from specific diseases. I'm working on the communicable disease branch. So usually you can think about splitting these surveillance systems into branches. You can think about either you have a new disease, right? And if one common question can be is, can we detect or even predict when a new disease might come, and how is going to be this new virus and where they are going to come? So this, we learn a lot from COVID. I know everyone started about COVID, but we did learn a lot that it's really important to have these systems ready in place so that we can intervene early and you can control. And then, so this is regarding pandemic preparedness. So I'm working 2 projects on that. And the other branch is about established disease. You already have some disease that are established, and the surveillance systems are there to track whether how fast they are growing, where they are going to appear, how they are going to strain the system, so that we can again have safeguards in place, and the public policymakers and develop policies and interventions to control the spread of this established disease. And then I worked on one of the projects is regarding the second branch. So my projects is the first 2 are related to avian flu. So avian flu, as we all know, is a potential pandemic endemic virus. It's zoonotic, so they are usually in wild birds. They are found in wild birds, and then we have spillovers from in poultry farms. We had spillovers in dairy farms in the U.S., and we even have a human case here in BC, unfortunately. So the idea here of these 2 projects is that there are lots of questions, open questions regarding even flu. And here we are trying to assess the risk of spillover from wild birds. So trying to understand how the virus in the birds are going to be, what are the chances of this virus jump to a farm or to another mammal or to human, and so on. And also trying to understand the virus itself. So the virus is always mutating. And then right now they are very efficient in spread with the avian. But maybe they can mutate and eventually become very efficient in spread with the mammals or with the humans. So the idea is like, can we get, so with so much technology in all the sequencing, whole genome sequence we have, the question is, can we use this data to predict whether a new virus, a new strain of avian flu might be very efficient in infect humans, transmit human to human. So these are the two projects regarding the pandemic preparedness. The next one is where I work with established disease like COVID, RSV, and influenza, so in respiratory disease, and try to understand what are the burden of this disease in the emergency department visits. So in how they burn the hospitals, right, the emergency departments. So I'm going to first start talk about the white bird risk project. So this project, we have the assumption that where you find a place that there are a lot of birds, it means that you have high chance to find birds that are infected, right? So the idea here is to map in BC or to have a map that's going to show the areas where we can have you can expect the high count of white birds, and not every white bird. We are focused on species that are going to be relevant to avian influenza. And then once we identify this hotspot, this can be used as a proxy for risk, right? That will be the next step. How can we use this information, the expected count of the birds, to then have an estimation of the chance of encounter or be exposed to avian flu. So the data source that I'm using these projects is all open. So one of the data sources is actually really fun. Data source is eBird citizen data. So I don't know if there are any bird watchers here in this talk, but if you are a bird watcher, usually you go watch birds, and then usually you input your data and your observations in this data, in this website, in this app, and then I can use this data to inform my model. So basically, I'm going to use this eBird that's going to tell usually in this data, they say the location, how many birds of each species are person observed. And I'm also going to use environmental data. And the idea is that the model is going to leverage these two data. So the model is going to look for places where you have characteristics in the environments that are going to be influence the bird decision to go in that area. For example, is there any food in these regions that the bird can forage? Is there like places they can nest? Is there any wetland or water, if it's like a waterfall? And also using the eBird observations as a way to have an estimation of the counts of birds that you can find there. And on the right side, I have an example of an output of this model that we are going to have. So for different seasons, we are going to have different distributions. And the idea, we wanted to find the true distribution for each season, right? The true count of birds around these regions in BC. So this project, we had lots of challenges, and the main one is the data is really biased. And the first thing is that people don't go everywhere to watch birds, right? They go to the places they either like or it's nice to watch. So there are a lot of areas that we don't have information on, we don't have observations. So the data is really biased, and then it's really nice. We have a strong need to understand the underlying process of the data and the underlying process of people going and watching birds. And also we need to, when you think about the environmental data, that we have to decide which data we are going to use. Are we going to use temperature, elevation, wetlands, distance to coast, how to make these decisions? We need to understand the biology of the birds, right? How the birds are going to make the decisions, where they go, where they are going to look for food, and so on. So we need to have strong collaborations and build relationships with ecologists and veterinarians so that we can bridge these two knowledge together. The model itself, it's when we are working with modeling and we think about your modeling for decision systems, for the end users are going to use our output of the model. Is essential that the model, we know the limitations of the model, and we explain that clearly so that people can actually have an interpretation of the results. So the main thing I always like to remember this model is that we are making an assumption here that high areas with high abundance of birds means that you have a higher risk to encounter infected birds, and that might not be necessarily true. So that's another thing that is a limitation, and something that we need to think about how to model in the next step. And then I think the lesson learned here is like we're collaborating with the Ministry of Agriculture in this project, and they are doing they also do lots of work and research on this on avian flu. They have lots of interest to understand avian flu because of the farms in the sea. And I think one thing, this project was delayed a bit because we had lots of difficulty to have to make sure that the projects weren't overlapping, in the sense that we need to be very transparent and clear. What are the goals? Like, and also be clear that you are flexible, right? To adapt. What are the needs? What do you need? So it's such an area that there's so much unknown that's really hard to know what's is the main thing needed. But that's how we end up defining the project as the whitebird ambulance, because the first layer is the very bare basic need that they have right now. So the next project that I'm working is the genomic risk. And here we are trying to understand the host transition. So it's like how a virus can identify in a sequence of the virus, whether that virus can transition from host, like instead just infect avens, one does a virus can start infect humans. So that's the main question that you want to ask. And then in this project, we are using, so you have, when you have your sequence of your virus, you have your genomic sequence, and the genomic sequence that's going to give you like the DNA, right? The RNA, and then that can be translated into the protein, the amino acid sequence that's going to give the proteins that you are encountering that virus. And basically, the proteins, they are going to be responsible for functions of the virus. And then we want to see, can we identify, if you get the sequence of proteins, all the amino acids, can we identify whether that virus is going to have a better time of infect humans? And then to do that, we are going to use, we are leveraging the protein language model. So in terms of generative AI, and these very large models that are trained with lots of data, so we are trying to think whether this can help to identify these patterns in the strains of new virus, new avian flu. So right now, this model is under development, and I think the main challenging here is that not only the data is complex, we have this very large database with a large sequence of proteins. We also have the model that's really complex as well. And then when you are dealing with modeling with so many complex and pieces and so many parameters, what can happen is that you can have very good results, but that doesn't mean the model is working. It just means that you have a really problem in the way you are, is the architecture of your model, or in the way that you are having assumptions in your model, or in the way that you are dealing with the data. So the data process is really complex. And as I said before, I'm a mathematician, so I don't know, like the part of the biology is not my expertise. So I really need collaboration with bioinformaticians because they have the knowledge of how the sequences are made, how the sequences are treated, in the alignments. There are so many things that they do before from the measurement process until the data get to me. And all of these steps are important to know so that we can see whether the model is being good, because we are developing a nice model and is actually learning the biology between these decisions of of the host of the virus, or just because by chance we organize the data in a way that the model was just memorizing. So here we need lots of communication, and the biological interpretation is really uncertain. So you need to be very careful when dealing with models like that and communicate the results. And the final model I worked on was the one on the public health systems forecasting. So this one was, I'm working with 3 pathogens now, with RSV, influenza, A, and COVID. And the novelty, the new thing of this model is that I'm modeling these 3 diseases at the same time and use different data sources. So I'm using wastewater, viral load, and reported cases, and respiratory emergent department visits. So I'm using all these different data streams together to model, let's say, the true infection distribution of these 3 diseases, so that with that I can have an attribution. I can, at the end of the forecast the respiratory emergent department visits, and also attribute how many of these visits are due to RCV this month, how many of these visits are going to be due to influenza in this other month. So on the right side, you can see the results. And then this model also accounts for cross-regional interactions. And this is really important when you think about respiratory disease, because people, they don't necessarily live in the same place where they work. And this mobility they can transmit in different places. And it's important to take into account when you think about the burden of disease and this transmission of disease across different cities. So the main challenge of this project is that the operational side, right? We have different data streams, so they are all going to be updated in a different time. The operational timelines, they are going to have a misalignment. We are going to have some noise in the data. So we are going to have, for example, ED visits. The data is always updated. So the last endpoints of your data is going to be updated in the next update of data. So that's not going to be necessarily the true value of emerging department visits. So this was one thing to take in consideration when developing this model. And the other thing is communicate uncertainty, right? Because the idea is that this model is going to be used by the health system so they know how much volume they're expecting for the next month of visits in the apartment, and what are the main diseases that are going to be affecting these people. And then we don't give you just 1 point estimation, we give like a range, so they have an idea that is going to fall within this range. And in the second step that we made sure is that I think it's very important is to have a reproduce. When you are working with modeling and developing softwares, we want to make sure that other people can easily use and apply that project. This is now a way to double check your work, right? If someone else get my package and they use, and in that scenario something is failing, so this is a way that I can improve my model. So we made sure to have this open so everyone can go in the website, in BCCDC website, GitHub repository, and access my package and use it with their own data. So my summary is that I'm kind of repeating myself, but communication is essential for this embedded research. I had the chance to work, as you saw, with different service lines, people with different backgrounds, and we are all trying to answer the same question, but using our own perspective. So it's really important to communicate, to be clear, and so that we can understand what are the needs, and we can tell whether a model can answer or not the needs, because sometimes a model cannot. So that's really important to have these communications, the limitations of your results. I think bringing back on what Katrina explained, discussed a bit between different academic research and embedded research. I think for me, when I'm doing academic and I'm developing a model, I'm answering a research question, right? Something that I define in the beginning, and I do the model for that. And here is a decision-oriented model. So you have to think about what are the operational logistics here? Like, how often do you want this result? What do you need? Like, what is the, do you need any uncertainty around this estimation? So it's really about the needs of the institution. So you have, sometimes you don't focus on how cool you want a model to be, it's just how useful that the models needs to be. And that might sometimes make you develop models that is not actually very cool, and sometimes you get do very nice models as well. The third thing that I wanted to emphasize here is I don't think I had ever experienced the chance to work with people from such a diverse backgrounds and skill sets. I work with MEDs, I work with veterinarians, and public decision makers, and microbiologists, and you get to learn so much. I think I never knew about like bird ecology or wastewater treatment. When you think about the how they are going to sample the wastewater and how they are going to measure the viral load. So there are things that I got to learn just because of this fellowship. And I think the fellowship itself, this my fellowship in my project, they are trying to address a very common public health challenge. That's when you think about disease, surveillance systems, infectious disease, is that we never know how many true infected people are there. We just know what we see. That's when people go use the health system and they test, and you see that they are sick, they test positive. So the surveillance itself is going to be incomplete, right? So we needed to think about how models can help decisions be made in real time with incomplete information. So that's it, and thank you everyone. Here are my mentors and collaborators that I have worked, and thank everyone for listening.